guys, welcome to our channel Biology Buddies. In today's video, we will be discussing one of the topics of Chapter 7, the DNA replications. So, let's start by exploring why DNA replication is called a semi-conservative process. So, semi-conservative is a term that describes the pre-existing strands of DNA will act as their template for synthesizing new strands of DNA. Hence, their new DNA will consist of one old strand and one new strand. So, now the process we'll start with the unwinding and unzipping of DNA via the helicase, breaking the hydrogen bonds that connect the two antiparallel strands of DNA together. By doing so, the helicase creates the replication fork which runs in this way, and then the topoisomerase will reduce the twistings or the positive supercoil created by helicase. And one of the type of topoisomerase is DNA gyrase. After that, the SXB proteins, which we could call the single-stranded binding proteins, will bind to the two separated strands of DNA to help stabilizing those two strands. And the SSB will help to prevent their strands from reconnecting or reannealing. And keep in mind that the SXB will be discarded once a new DNA molecule is synthesized, or we could say at the end of the replication process. Moving on, their DNA primase will generate the primers, or we could say produce the primers, these yellow rectangle here that will serve as the start here point or the starting point that is attached to the two template. This one attached to this one, and this one attached to this template strands. And then these primers will send a signal to the DNA polymerase tree to tell it where to start synthesizing the new strands. And so once the RNA polymerase tree bind to these primers, it will start to synthesize their new strands by adding the nucleotide covalently to the newly synthesized strands via phosphodiester bonds using the old DNA strands as their template, which means their nucleotide from their new strands will be complementary to the nucleotide from the OR template strands. And one of the most important informations that we should notice is that their DNA polymerase tree run in the directions of 5' to 3' always, and according to the anti-parallel rule, which state that the two strands within the DNA molecule should run in the opposite directions. Hence, on the leading strand here, which run in the directions of 3' to 5', their DNA polymerase tree should move along with the replication fork this way and synthesize the new strands in that directions to satisfy those rules that I just listed, and this leads to the new strands being continuous. While under lagging strands, which run in the directions of 5' to 3', due to the anti-parallel rule, DNA polymerase 3 will move away from the replication fork. Therefore, the new strands will be synthesized discontinuously forming Okazaki fragments. Then, later on, so DNA polymerase 1, this green circle here, will replace all the primers with their DNA nucleotides. And for the lagging strands, their DNA ligase, here the DNA ligase here, the 31, will join the Okazaki fragments together by covalently joining their superphosphate backbones between their nucleotide after fragments with a phosphodiester bond. And then after all, we will get the two newly synthesized strands of DNA, and the cycle will continue to produce hundreds and thousands and even millions of new DNA molecules. Yeah, and so I think that is the end of the DNA replications video. We hope that this helps you to understand the DNA replications a bit better, and you can contact us for further informations using the socials links in our bio below. And thank you guys. Good luck on your journey of grinding the biology IB. We are all in this journey together. And thank you, and hope to see you guys again in the next video. Goodbye for now.